Okay, so we just got done talking about regulation of DNA replication on bacterial chromosomes. There's just a couple other things I want to talk about before we start talking about the next step in macromolecular synthesis, okay? So one is how we replicate DNA in the lab, because this ends up being really important for research and a lot of other practical applications. This is called the polymerase chain reaction, and I encourage you to click on this link to watch a video of how PCR works. So PCR takes advantage of having purified DNA and a purified DNA polymerase. It's called Taq polymerase from a bacterium called Thermus aquaticus, which is very thermally stable. So in a PCR reaction, as you can see here, we use heat to split the strands apart. Then we have short synthetic primers. So this is sort of taking the place of what DNA primase provides in the bacterial cell. These DNA primers will bind to their complementary sequences on the isolated DNA, and then the Taq polymerase will use those as the starting points to extend synthesis. And if you go through cycles of this, where after each time the polymerase extends, you heat the mixture up and melt the DNA, separate everything apart, let new primers come in, then each cycle you're exponentially increasing, you're doubling the number of copies of that specific DNA sequence, which is bounded by these two primers that you have. And if you do that 30 times roughly, then you'll get about a 1,000,000-fold amplification. Okay, so this is important for a lot of different applications that I'm sure you've heard of. I just wanted to make sure you were aware of that. If we were actually doing a lab in micro this year, then we would do some PCR. But I just want to throw this in there because this is a synthetic version of DNA replication. The other thing I think I wanted to talk about for a completely different reason is plasmids, because plasmids are not part of the chromosome. So what we were just talking about with OREC and replication of the chromosome does not apply to plasmids. Plasmids will have their own origin and replication. This example here, PBR322, is actually a synthetic plasmid that was made through recombinant DNA technology, but every plasmid, whether it's natural or synthetic, has an origin or replication on it that carries out an analogous role to what OREC does, although the precise mechanisms of replication and how that origin is used can actually vary from plasmid to plasmid. It's not always bidirectional replication. There's another replication, or at least one other replication mechanism called rolling circle replication that applies to plasmids. But the key thing to remember is that a piece of a plasmid DNA is a separate DNA molecule from the chromosome in a bacterial cell, and thus it has to be replicated separately. Okay, usually, well, pretty much always, they're much smaller than the chromosome. They're usually circular, although there are rare exceptions of linear plasmids, just like there are some rare exceptions of linear chromosomes in bacteria. And they do need host proteins to replicate, but they often provide some regulatory functions controlling their replication themselves. Okay, so here's what I was just saying: bidirectional replication or rolling circle replication. There's different mechanisms. There are a number of copies of a plasmid in a bacterial cell can vary. Some of them keep their copy number very low, comparable to the chromosome, or a little higher. Others keep their copy number pretty high. So there may be 10, 20, 30 copies of the plasmid per cell. The copy number also affects how they segregate between daughter cells. A low copy number plasmid, like a chromosome, has mechanisms to actively ensure its distribution to the daughter cells after or during replication, whereas a high copy number plasmid may just rely on random chance. If you have enough copies, it's highly likely that there will be some copies in both of the progeny cells. Plasmids have a number of biological functions. They can have genes that provide things like resistance to antibiotics and heavy metals. They can be involved in pathogenesis, in symbiotic relationships with host cells. These are things that I'm asking you to think about in the lab this week when you're going through a paper on the genome of a particular pathogen. If it has plasmids, and I want you to, and they think that they're involved somehow in the process of attacking a host, for example, then I want you to talk about that, okay? And then finally down here is that plasmids, conjugal plasmids at least, have mechanisms to transfer between cells, and that is something that we will again talk about when we talk about horizontal gene transfer in the separate lecture for the lab this week, all right? Okay, let's talk briefly about what a gene actually is, and then we'll talk about the next step after replication, which is transcription. A gene is a region of DNA that encodes a functional RNA molecule. Now, we usually think about messenger RNAs because those encode proteins, but there are other functional RNA molecules like transfer RNAs that carry the amino acids to the translational machinery, ribosomal RNAs that actually help make up the translational machinery, and then there are a number of regulatory RNAs that are highly variable from organism to organism, okay? All right, so now we're going to talk about the process of transcription. Now, one of the things I want to just show you on the board right now is how we're sort of organizing this. We're talking about what is often referred to as the central dogma of molecular biology, right? So we have DNA is our primary informational molecule, right? This is the basis of the hereditary functions in an organism. DNA is copied into RNA, which is then used as a template to make proteins from, right? So this is often referred to as the central dogma of information flow in the cell, because this process is referred to as transcription. This process is referred to as translation. And each of these is a way of transforming the information that's present in these molecules. And then, of course, DNA, you have to make new copies of that, right? So we can just write a circular arrow there, and that we refer to as replication, right? So this is referred to as the central dogma, D-O-G-M-A, of information flow in cells or in organisms, right? So replication, transcription, and translation, DNA to RNA to proteins. The primary violation of this that we've talked about a little bit in this class, and we will talk about a little more, is the fact that in retroviruses, we actually have a reversal of this, or they have an RNA genome that is reverse transcribed into DNA. But reverse transcription is not a common process in biological systems. So generally, the information flow goes like this, okay? So we talked about replication. Now we're going to talk about transcription, and then we'll talk briefly about translation. Hopefully that red color came through when you're seeing this. Okay, so if a gene is a functional RNA, is capable of producing a functional RNA molecule, transcription is the process by which it does it. Again, there's a nice animation here. You can click on this and see this much more visually and dynamically than what I'm going to just tell you about it here, right? So initiation is the process by which transcription starts at a promoter. A promoter is a sequence on DNA that is recognized by this key enzyme, RNA polymerase. RNA polymerase is the enzyme that makes the RNA molecule from a DNA template, okay? So the RNA polymerase, it's shown here as this tiny little molecule. In fact, it would be much bigger than this, right? So this is not a 2-scale diagram, unfortunately. This area here, just like we had a replication bubble, you might think of this as a transcriptional bubble. But unlike in DNA, where that replication bubble is never going back together, in RNA synthesis by RNA polymerase, this bubble is only transiently open. And after RNA polymerase leaves the promoter, those DNA strands will come back together. Okay, so this is going to be a moving bubble. It's not going to be permanent, right? So RNA polymerase is going to separate those strands of DNA so that it can access the template strand. It will then lay down a complementary strand of RNA using the DNA strand as a guide, right? And it's going to move in the 5' to 3' direction relative to the RNA that it is synthesizing, which is going to, of course, be anti-parallel to the template strand. Okay, so the template strand's like this. The RNA molecule looks like this. The RNA molecule sequence is going to be equivalent to what we call the coding strand here. The only difference between those sequences is going to be that the RNA polymerase is going to put U, or uracil, or uridine, if we're talking about the nucleotide, in place of the T, or thymine, in the DNA. So RNA only does not have thymine, it has uracil in it, okay? So it's the equivalent sequence to the coding strand, the complementary sequence to the template strand there, okay? RNA polymerase holoenzyme has a couple of different components to it. The core enzyme is shown here, has bound to it a subunit called the sigma factor, which turns this into the holoenzyme. I wanted to show you the structure of the core enzyme. It has a couple of subunits called alpha, of which there are 2 copies, beta and beta prime, okay? So this is the core RNAP, that's the abbreviation for RNA polymerase. And then to that, you're going to add the sigma subunit. The sigma subunit is going to identify the promoter, and it's only there at the start, okay? So with sigma bound, you recognize this promoter region. The promoter region is a specific sequence of DNA, or at least it's specific in this so-called -35 and -10 regions. In between there, it doesn't really matter. Sigma is going to bind to that and say, hey, this is a promoter. This is where we should start transcription. Without that sigma subunit, RNA polymerase has the ability to make RNA, but it doesn't have the ability to recognize a promoter. So it doesn't know where the genes actually are. So the holoenzyme, which contains the sigma, finds a promoter, and then it separates the strands. The sigma subunit doesn't, but the RNA polymerase does. And then it starts synthesizing RNA, at which point the sigma factor isn't needed anymore, so it comes off. Okay, down here at the bottom of this is a diagram of what the promoter actually looks like. Up here, remember it said -35, that's this sequence here. -10 is this sequence right here. Okay, so this characteristic sequence of the dominant sigma factor in E. coli, at least, and related bacteria is shown here. This varies a little bit in bacteria, but that's what the sigma subunit is recognizing, okay? And then the core enzyme starts to synthesize the RNA, sigma comes off, core keeps going until it gets to the end of the transcribed region. We'll get to that in just a second here. It is possible to have other kinds of promoters than the one that I showed you there, right? This promoter sequence is recognized by the dominant or sigma factor, which is also called sigma 70. So sigma 70 equals the dominant sigma subunit in E. coli, at least. Okay, so most genes use that 70 kilodalton sigma subunit, but there are other types of promoters that use different sigma subunits for specialized functions, right? For example, the sigma 54 is for nitrogen utilization genes. Sigma 32 is for heat shock response, etc. Sigma 28 is involved in the expression of flagellar genes, and that's a story we'll get to next week, right? So if heat shock genes, for example, since they're all going to be transcribed by Sigma 32, they're actually going to have a different promoter sequence here than the one that's shown. So this is a strategy for controlling whole sets of genes. This is one strategy for doing that, okay? I don't care if you remember the details of which sigma factor is used for what set of genes, just remember that it is possible to have different sigma subunits that can recognize different promoters, okay? Now what happens, how does the RNA polymerase know when it gets to the end? The most common way of doing it is that there is a sequence after the last gene that is transcribed. It's called a termination sequence. This is actually for what's called row-independent termination. And when the RNA polymerase reads through that, it makes this stem loop structure, and then there's a sequence of AU base pairs there that would hold the transcript to the template, and AU base pairs are the weakest possible base pairs, and so the formation of this stem loop and the weak base pairing there is going to cause this complex to fall apart. Okay, so a terminator has to come at the end of the genes to be transcribed. Okay, so there are some important differences. We're not going to go into the details of eukaryotic transcription. Just want you to recognize that there are some important differences here. One is that bacterial genes don't have introns, so there's no process of splicing in bacterial messenger RNA or RNA in general. Another is that the 5' end of the RNA is a little different. There's a cap added in eukaryotic transcripts that's not present in bacteria. Another important thing is the presence of a poly A tail in most eukaryotic transcripts that's added post-transcriptionally to tag this RNA. That is not present in bacteria as well. So no poly A tails, no introns, okay? Okay, I am gonna stop there for now and then pick up with another recording.